Philosophers and scientists have wondered for centuries on whether or not experiences have an impact on the physical change in the brain. In the 1960s, we finally gained the ability to measure brain development, leading us to our experiments. Mark Rosenzweig came up with an idea to test this experiment on rats. Using the hypothesis, animals raised in more stimulating environments will show a different brain development than those that are in a duller environment.
3 male rats were chosen from each litter. They were then randomly assigned to one of the three conditions. One rat remained in a laboratory cage with the rest of the colony, the enriched environment cage, and the third was assigned to the impoverished cage. Remember, 12 rats were placed in each of these conditions for each of the 16 experiments. The impoverished environment was a slightly smaller cage isolated in a separate room in which the rat was placed alone with adequate food and water.
The standard laboratory colony cage contains several rats and an adequate space for food and water always available. The enriched environment was virtually a rat's Disneyland. 6 to 8 rats lived in a large cage furnished with a variety of objects with which they could play. A new set of playthings drawn out of a pool of 25 objects was placed in a cage every day. Our results showed that the brains of the enriched rats were different compared to the impoverished rats in many ways. The cerebral cortex of the enriched rats was a lot heavier and thicker. There was no significant difference that was found between the rats that had to do with the brain cells, but the enriched rats did produce larger neurons. Related to that, the ratio of the RNA to DNA, the two most important chemicals for cell growth, was greater in the enriched rats, meaning there was more chemical activity involved in enriched rats' brains. Most brain activity occurs at the synapses, when two neurons meet, when a nerve impulse is either passed on to the next neuron or if it is stopped. With this, the research found that the synapses of enriched rats were 50% larger than those impoverished rats, potentially allowing for greater brain activity.
So, we can compare the results in rats to humans. The environment our children are raised directly affects their brain functions. Because experience in our environment play a part in our brain functions, this information can be used to shape the next generation's lives. This gives us all the responsibility of creating a healthy environment for the youth and for each other.